Morning, everybody. Welcome to Gout, Symptoms, Causes, and Treatment. Hi, I'm Dr. Pete. I have a PhD in biochemistry, and I'm also a nutrition network health practitioner, and I specialize in the area of gout, but the main thing that I do is focus on fructose uric acid metabolism, how it relates to our other important metabolic pathways, primarily glycolysis and its connection to the mitochondria. And the reason that I focus on this is because fructose uric acid metabolism is one of the central mechanisms that's driving metabolic diseases that most of us are faced with as we age into our 60s. Diseases like type 2 diabetes, obesity, cardiovascular disease, Alzheimer's, as a matter of fact, and also sugar and alcohol addiction. You can think of me as a biochemical analyst. I am not a medical doctor. And as far as biochemistry goes, I've been at a point in my life, I do not have a laboratory and a team of scientists that work with me in order to run experiments, but I analyze the literature. And the way that I got into this 5 years ago was that I was diagnosed with prediabetes, and also I was a gout sufferer. And back then I didn't understand the connection between those diseases. And in interacting with the health establishment, my doctors primarily came back and sent me an email that said, you have this pre-diabetes diagnosis, go on the Mediterranean diet and get more exercise. And the problem that I had with that, one of the central problems was that at that point, I was a third-year CrossFit athlete. I was in the gym doing these really intense workouts 5 to 6 days a week. And getting more exercise, quite frankly, was not something that I felt like I really needed to do. Although, in response to that email, I did exactly what the doctors were asking for, and I did increase the amount of exercise that I was getting, and I did shift to a more rigorous Mediterranean diet. However, I saw my condition getting worse with time, my fasting glucose and so on. And when I went into the literature, mainly the medical literature, I discovered that basically what they were offering me was just future medical care and suggesting that these diseases are progressive, that there's nothing you can do to reverse them, that they're just going to now be part of your life. But if you do these lifestyle interventions, that you can slow the progression down. Now, as a biochemist, I looked into this and I thought about this, and I understood that if I was dealing with a metabolic disease, that this would necessarily have to do with what I put in my mouth. And so I started to look harder into the literature, and I found my way to Virta Health. And Virta Health, at the point where I got involved with their science, reading their papers, was that you indeed could reverse diabetes by going on a low-carb lifestyle. At that point, I hadn't really gotten into addressing the gout. It was in this shift to the low-carb lifestyle that I saw my uric acid rising significantly. And I became concerned because the question that all of us have that are hyperuricemic, high uric acid, and also in the low-carb lifestyle, and we see this significant rise in uric acid, we ask the question, what does this mean for me? Am I going to suffer gout flares down the road? And what's the relationship between the low-carb lifestyle, for example, and the fact that this uric acid is going up? And eventually, once I keto adapted, or I adapted essentially biochemically to eating less carbohydrate, I saw eventually my uric acid come down. However, when the uric acid comes down, we remain, for the most part, this is coming from my coaching experience over the last 5 years and working with a lot of gout sufferers, that we tend to, upon keto adaptation, our uric acid comes down to still a hyperuricemic range. And in the process of all of this, I ended up coming onto Dr. Richard Johnson's body of research, which is over 700 publications in the area of fructose uric acid metabolism and decades of research, both utilizing tissue culture models, animal models, and clinical studies with humans. And it was coming to that body of research. And now in 2023, there are a lot of other scientists now that are looking at different angles of fructose uric acid metabolism. And this is what allowed me to make this connection between gout and diabetes, obesity, and all of the rest of these metabolic diseases. Fructose uric acid metabolism is a central driver of metabolic disease. And in today's talk, we're going to focus on gout. But even if you're not a gout sufferer, you should be listening up to this, because we all are suffering from the same metabolic driver. Ultimately, how we respond to this driver as individuals is going to depend on what our collective genetics, I say collective because we're talking about in higher animal cells. In humans, for example, our genetics are mainly gene families. So if you, for example, do a literature search and ask the question, how many genes are involved in type 2 diabetes, you're going to get the phone book. There's a lot of genes involved in this. And that's because the way we respond to our food environment, our activity environment, the amount of exercise that we're getting, the level of sleep and sleep disturbances that we're having, potentially stress, all of those things contribute to what our consequential outcomes are going to look like. And we don't all have the same consequential outcomes. One outcome for me was the prediabetes and the gout. My gout is in remission now, and I reversed my prediabetes. Other people will have a different set of outcomes. For some of us, we end up being obese, and maybe there's type 2 diabetes overlapped into that, and maybe even also cardiovascular disease. For others, and again, this comes to what we call phenotype, my phenotype, right? Which is I appear to be, even 5 years ago, if you took a picture of me, you would see that I was overweight. Now I've lost 33 pounds and I'm weight stable. But my point is that back in those days, most people that would look at me across the table would say, well, you must be active, you're thin and all this. But I was, as we would say in my field now, I was fat on the inside. And this has a lot to do with the resulting dysfunction at the level of the liver. So with that introduction, I don't care why you're here. If you're a gout sufferer, this is a really good place for you to be. But if you are also a non-gout sufferer, but you are also suffering from other metabolic dysfunctions, this is a really good place for you to be this morning, because we're going to talk about the central mechanism. And with that, let me go ahead and get started with this. So what differentiates me from the rest of this space is that for most of us, right, most of the scientists and the medical doctors that are out there in the low carb space and trying to look at how we reverse these dysfunctions, they're standing on the outside of a forest. And that's the purpose of this slide. So there's a lot of ways to look at this stuff. My scientific training goes way back, and I was involved early on at trying to understand the gene expression in humans at the level of RNA. Now, I know that that sounds really abstract, but there are lessons from science, how we understand complex processes. And I'm here to tell you right now, it's very limited what we can learn if we stand outside the forest all the time and shoot arrows inside that forest looking for central mechanisms, right? If you're lucky, the arrow will hit the right tree. But more than likely, look at the number of trees, the arrow is going to end up embedded somewhere where we think we might be learning about something. It's super crucial in this business for us to go inside the forest. We have to go inside the forest. And if you track back any sort of serious biological science that's led us to the understandings that we have today, like even if you think about DNA and the structure of DNA, where did all of this start, right? It started with getting the crystal structure of DNA, that very basic knowledge, and then working out from there. And in my area of science, when we talk about how you take a gene at the level of DNA, how that gene is transcribed, for example, how we produce an RNA from it, all of that started in a test tube with very small pieces of RNA that we took out of other systems and put in the test tube. And then we beat to death what the process was in order to make RNA from this piece of DNA that we put in there, and later pieces of RNA that we put in there to see what would happen to them. And things happen stepwise. We understand in the test tube how something works functionally. Then we take it to the next level, right? Which might be actually looking inside human cells. And in the work that I did, that was yeast and that was HeLa cells. This was a tissue culture line that came from cells that were removed from a cervical cancer patient. And then eventually that work was extended out into other cell lines. And then eventually it was extended out into animal systems. And then it was extended out into eventually clinical studies, right? And this is how we came to understand how basic cells function. So a crucial thing that I feel, or piece that I feel is really missing in the area of metabolic diseases is fundamental understanding of how our cells are functioning on a metabolic level. And I think if you look at the history of this and you go back into the early 1900s, we were on the right track. This is what where Warburg came from, studying nuances of the glycolytic pathway, and then later, and how that was related to what was going on in the mitochondria. And then later we had Krebs, and then even later we had this guy named Veitch. There was a lot of really good metabolic science that was going on earlier, right? And then we've lost track of this. And for whatever those reasons are, some of it had to do with the discovery of DNA and a shift to trying to understand on a molecular level what was going on there. But I'm here today to tell you that what I do primarily is I look at the literature. I was trained as a biochemist to turn over every single rock. I don't believe in staying in my lane, right? If you want to solve a problem, you have to look at the science and you have to go where the science takes you. Learn from it what you can, ask new questions, and keep moving. And be prepared to be wrong. So today, and the reason we need to be prepared to be wrong is because we want to solve this problem. And when there is a hypothesis, if that hypothesis turns out to be incorrect, we can't still stay banging away on the hypothesis as it was sitting there. When we get a new piece of data that suggests that something that we were thinking is incorrect, the hypothesis needs to be rewritten, and then we need to focus on that, doing the experiments to either prove or disprove it. And when we get data that shows that that something else in the way that we think is not correct, then we have to change the hypothesis and we have to keep moving. If we want to understand why people like me at 60 years old are showing up with diabetes, the comorbidities that come with obesity, the cardiovascular disease that is leading to people getting stents and falling dead, then we need to understand what's going on at a core metabolic level in order to make changes to our lifestyle that makes sense and can reverse those conditions. Now, if we turn to gout and we ask, okay, what is it? This is a specific inflammatory arthritis. I believe when you compare this arthritis to others, that all of them involve some level of inflammation, even osteoarthritis. I don't believe that splitting these arthrites up into categories relative to whether or not they have a certain kind of inflammation is really that helpful to us. We need to understand that all of them involve inflammation on one level or another. And this suggests a core driver on an intracellular basis within the biologically important cells that are actually in the joint. The nuances come into play with how that intracellular inflammation is playing out into the extracellular environment that is around those cells. And then how that extracellular environment is predisposition because of our genetics to go one way or another, and then where the outcome is with the swelling and the discomfort and the pain. In the case of gout, we're talking about the formation of uric acid crystals that interact with the innate immune system to generate the gout flare. In rheumatoid arthritis, there looks like, and I'm going to touch on this as we go through this, there is also a connection to uric acid. The difference between rheumatoid arthritis and how it's interacting with the innate immune system and perhaps gout is the gouters, that's me, predisposition to form seed crystals. That could be what the difference is. Usually gout affects a single joint, but in 2023, we know of cases that involve symmetrical joints. And when I use the term symmetrical, what I mean by that is somebody could have gout in the major joint of both toes. That is possible. Most of the time, it's a single joint. Or it could be you have gout in one knee or the other, or one finger joint versus the other, or it can be in multiple joints. Gout is an extreme debilitating pain. I've experienced it myself. I've had flares more than once. The secondary flares that I had later were because I went on urate lowering drugs, and I may have time to get into that subject area a little bit later in the presentation. But here's the key thing. If you ask a gout sufferer to rate the pain from 1 to 10, they're going to tell you it's a 10 every single time. And I know that when I had my very first flare and I got out of bed and I stood up, I literally nearly fell down on my face because the pain in my foot was so extreme that I honestly thought I had broken my foot. Gout is treated like it's an inconvenient illness by the medical establishment. When you go in, because usually people don't realize that they have gout. Like I said, I got out of bed, nearly fell on my face. I went to the doctor thinking I broke my foot. That's when I found out I had gout. And usually, and I went through this experience, once you've discovered that you have gout, then what the doctor's going to do essentially is give you a lecture about lifestyle. You are stigmatized. You are treated like you're a glutton and a victim of excess. And you're going to be told stop eating that way. And if you are drinking alcohol, they're going to ask you to stop drinking alcohol, which is actually a really good thing. But my point is that they're going to treat you like it is that you are a person of excess. And truth of the matter is, when you look around you in a family setting, extended family setting, going all the way out into your friends, they're all eating exactly the way you are eating. The difference is their genetic predisposition is different than yours, and that's why they're not popping up with gout. But I'll cite the same observational science that most of my colleagues are talking about on the internet all the time, and that's that 88% of our population is metabolically unhealthy. And you can look at the data yourself. So basically, being treated this way by the doctor is not very helpful. And the stigmatism is real. I mean, once I knew I was a gouter, I didn't talk about it for a long time. And in fact, at work, when I was limping around, I told people I had, sprained my ankle. I didn't want to talk about the fact that I had a gout flare, because this was something that, it came out of left field, and I was made to feel like, I was some sort of when you look at the history of gout, like I was some sort of a king or prince that was sitting around in a big room at one end of a long table, eating desserts all day long and drinking sugar fortified wine. And that just wasn't the case. I mean, I was guilty of eating a high glycemic meal. I will talk about this coming up. Hyperglycemic meals and drinking 3 beers every day. I was doing things like that. But again, when you look around and you compare yourself to the other people that are out here in our culture, my eating plan wasn't very much different than theirs. And in fact, the more people I know who are in my age range, every single one of them is suffering, mostly who have not made lifestyle changes are suffering from metabolic diseases that are being driven by the same central biochemistry. Remember, going into the forest and looking at the nuances of our metabolism. So when we boil down to it, this is the definition of gout by the medical establishment. I don't care where you go to look this up. Any of the major associations, they all essentially have the same definition. And what that definition says is that we have a rise in uric acid in the circulatory system. That uric acid can diffuse into the joint. When it does, it crystallizes. They give you this sense that the crystallized uric acid has sharp little points that are going to be essentially the idea, jabbing into the joint, causing this inflammation, which is the gout flare. I call this the sliver in your finger model. So going into this right now, the one thing that the literature shows, going all the way back into the 1850s, is that a simplistic mechanism like this is incorrect. I am not saying that uric acid is not required for a gout flare. It is required for a gout flare. However, it is not sufficient to do it on its own. And crystallized uric acid indeed is required in the flare, but this happens in a much more complex mechanism, and it's not about a sharp crystal. In fact, uric acid crystals are brittle, and they don't do well under pressure. They break apart into smaller crystals. This is all about how our innate immune system actually interacts with the uric acid, which I'll be coming to. So we can start out the presentation basically by outlining what I call the 3 myths of gout. The first is that uric acid is a waste product of protein metabolism. All right, if you look in a biochemical textbook and you look up the degradation of protein, you indeed will see uric acid at the end run of degrading the RNA and the DNA that is going to come out of this protein source that you're eating, like chicken or beef or lamb, fish, whatever it is that you're eating. Indeed, you can find uric acid popping out at the end of that. But here's the thing going into this that you've got to understand. Uric acid is a signaling molecule. I call it an electron transfer oxidation reduction molecule that is involved in networks with other molecules that are just like it. Uric acid exchanges electrons, and it's involved in regulatory networks, for example, in the liver, that actually signal a shift into fat storage, systemic inflammation, and modulation of nitric oxide, which downstream has significant influences and impact on the cardiovascular system. Uric acid itself is not sufficient to cause a gout flare. So this word, in terms of science, sufficiency, is super important, because going back to the last slide where we were talking about the medical definition of gout, that definition assumes that uric acid is sufficient on its own. There's nothing else needed besides the crystalline uric acid to cause the gout flare. So sufficiency is a really important issue here, and uric acid is not sufficient to cause the flare. There are indeed other factors, and there's more complexity to this. For example, soluble uric acid along with crystalline uric acid is required for the flare. And then the last thing is this idea that uric acid lowering drugs actually treat gout, and this is also a false narrative. Uric acid lowering drugs do what they say, they lower uric acid. But it is, in fact, the fluctuation of uric acid, either up, the acute rise of uric acid up, or the acute decline in uric acid down. It's that dynamic where a gouter is at most risk for having a flare. And the truth of the matter is that when you go on urate lowering drugs, the probability that you are going to have a flare in that process to bring the uric acid down from wherever your average uric acid level is to a new average lower, that in that process, there's a high likelihood that you're going to have a gout flare. And this says directly that a urate lowering drug is not decreasing the probability of a flare. What this means in the long run, and this may sound confusing to you, in the long run, getting to a new lower average, that in fact, if you hold ground on that new lower average indefinitely in terms of time, this can have the effect of lowering the probability of a flare. But during the transition of actually taking the drug, there's actually a higher probability that you are going to flare in that process. And this is because the urate lowering drug is not actually influencing whether or not you have the flare. It's this special inflammatory state which can exist regardless of the drug that is influencing in partnership with whether or not you get the seed crystals and whether or not you're actually going to flare. When we ask the question, so here's the thing, gout is not a local disease. Gout, it has a systemic component to it that is driven by fructose uric acid metabolism, and it has a local effect. And you can see that when you look at the statistical data. 3/4 of the people that are gout sufferers also have high blood pressure. Almost the same percentage is suffering from some level of kidney disease, and that's because fructose uric acid metabolism is active in the kidney, highly active there. Over half are obese. A full quarter of them are also suffer from type 2 diabetes. A full quarter usually will have kidney stones, and somewhere around 10 to 14% are going to be suffering some level of cardiovascular disease and or strokes. And those are all diseases whose central mechanism is being driven by fructose uric acid metabolism, in my opinion. When we look at the statistics for gout, we see that we're at about 4% of the US population, and that percentage is going up. 43 million of us are walking around with hyperuricemia. This is circulating uric acid, which is considered high. What does high mean? So the reference range, when you go in and you have your blood tested, is going to be between 4 mg per deciliter of uric acid and 8 mg per deciliter of uric acid. And hyperuricemia in men is taken to be 7 mg per deciliter or higher, and in women, 6 mg per deciliter or higher. So 43 million of us are walking around with what's considered to be hyperuricemia, but only a small percentage of that, relatively speaking, will be gout sufferers. So just having hyperuricemia alone, and this goes back to what I was saying about the medical hypothesis of gout, is not enough. It indicates that just the presence of uric acid, or even high levels of it by that the medical establishment has admitted to, is not enough to have a gout flare. When we ask the question, what is the average concentration of uric acid in a gout sufferer, the average is about 8.3 mg per deciliter, which is just at the upper range of the uricemia, the standard range in the laboratory. We see that a full 18%, though, have uric acid between 6 and 8. So that means that a lot of gout sufferers have uric acid values that are within the normal range, and a full 14% of them have gout flares under 6. And I'm one of those guys. When I went on a uric acid lowering drug, allopurinol, I had a gout flare where my uric acid was actually 4.5 mg per deciliter at a constant range, right? I had already been at a uric acid level for a matter of weeks at between 3 and 4 mg per deciliter, and then I had my flare. And then finally, and this goes back into data, really high quality data from the 60s to 70s and the 80s that showed that you could extract the synovial fluid from hyperuricemic people that were not suffering from gout and find the presence of uric acid crystals. So stepping back from this, from a systemic point of view, fructose uric acid metabolism has been shown not only via tissue culture, animal studies, and clinical studies, most of which has come out of Johnson's laboratory, over 700 publications, to drive not only gout, but kidney disease, diabetes, type 2 diabetes, obesity, fatty liver disease, Alzheimer's, the various permutations of cardiovascular disease, and sugar and alcohol addiction. So there's a strong systemic component to gout that has to do with the production of uric acid, and I believe this starts with the liver. The liver is the central driver of metabolism in a lot of different respects. Some would argue, well, there's got to be a strong interaction coming in with the brain, and they're not incorrect. I agree with that. But when we look at the liver and we ask the question, what is the relationship between fructose uric acid metabolism and glycolysis, there is very powerful integration of these pathways. And this is one of the problems when we're standing outside the forest shooting arrows into the forest, and we think about these metabolic pathways in an isolated way. First off, the pathway that I'm illustrating here on the left is glycolysis in gray. And glycolysis is connected to the mitochondria through the production of pyruvate and lactate, very important substrates that get transported into the mitochondria that then can be processed through acetyl-CoA into the Krebs cycle or the tricarboxylic acid cycle, depending on which name you want to go with. And the electron potential of that circular pathway, Krebs, is delivered into the electron transport chain where we produce ATP in large quantities. This is the energy currency of the cell. You can think of it being like molecular money. We need money in order to do things. The cell, when it's going to build this or build that, has to be able to utilize this ATP, the energy currency, in order to pay for those processes. And glycolysis itself, when it is operating under essentially low glucose conditions, the main purpose of it is to produce, we call it on a substrate level, some ATP there. And we know from biochemical studies, this is enough ATP to run the cellular processes that are going on in the background. When glucose rises to a certain point, we can also make a little bit of glycogen. And to stay consistent with the medical literature, I'm going to say when glucose rises to a certain point, albeit remaining within normal levels, we have not gone to a hyperglycemic level, and this is important, then maybe we produce a little bit of fat if the energy requirements of the cell in connection with the mitochondria are adequate for whatever the person is doing at that moment. Now, as glucose rises, however, there is a threshold that gets crossed into the hyperglycemic range. And in the scientific literature, understanding what this threshold is still, in my mind, an open question. We don't know what that threshold actually is. But what I'm going to tell you here is that once we pass that threshold, we open the polyol pathway in the liver. And this is a two-step biochemical process. Once it's opened, we're going to start funneling upwards of 30% of the incoming glucose into endogenous fructose that then is processed through the fructose uric acid pathway that results in an acute decline in the amount of cellular ATP within the liver, with an acute rise in uric acid, which is a signaling molecule, it very easily transfers electrons. It's part of this electron transport circuit, which is signaling a change. And in this case, the change that we have in the liver is a significant rise in systemic inflammation. We have the activation of de novo lipogenesis with the production of fat directly in the liver, resulting in oil droplets in the liver, and also the packaging of triglycerides into VLDL that are pushed out into the circulatory system. This is why when we do the bloods on somebody who is eating a lot of added sugar and hyperglycemia in their lifestyle, will have elevated triglycerides. This is the main reason. And we all have, and then additionally, we have the alteration of the nitric oxide pathways, which downstream in the endothelial layer of the cardiovascular system, which is altering a number of things, not the least of which is the elevation of blood pressure. And this is the reason, again, why somebody who's eating in a hyperglycemic way constantly, along with the added sugars and so on, and other factors I'm going to get to in a minute, have a high blood pressure on a chronic level. So to review this, and I believe I'm just going to cut to the chase right now, that when we're eating this way, where we violate this threshold and we go into the hyperglycemic state, we have added sugars coming in, perhaps alcohol also with the meal. We have processed food and processed meats that would bring in high levels of glutamate, which also activate this pathway, in inosine monophosphate, adenosine monophosphate, the umami effect, which contributes to opening these pathways. Then we drive, actually in the liver, a change in state from what I'm calling the burning state into a state that is characterized by storage, glycogen, fat, water, and an elevation in systemic inflammation, which has effects on the innate immune system, and disruption, at least at the level of the liver, on nitric oxide. But in other tissues where fructose uric acid metabolism is active, we also have modulation of the nitric oxide pathways. When we're doing this on a chronic level, we're eating this way every single day for most of our waking hours, then people remain in what I call now the red state. I believe we have a shift in states from burning to red. There are some influencers that are speaking out there in this space. Robert Lustig is who I'm thinking of right now, who talks about this difference in states, right? The burning state versus, I don't remember off the top of my head what he calls this state, but we're arriving at the same location, but starting in different places. The idea, again, going inside the forest and looking at the core potential causal mechanisms biochemically and at the level of molecular biology in regard to what's happening when people are eating the way that I'm describing here. So this is the first time that I've talked publicly about this. I've done some videos about my viewpoint of this biochemistry for the Nutrition Network, but this illustrates on a general basis this difference in states. So on the left, we have the burning state. When we look at ancestral humans, when we think about this from an evolutionary point of view, humans remained on a daily basis most of the time in the burning state that is characterized by low insulin. You know, going back to the slide that I had a minute ago, when we shift in the liver to the state that I'm talking about, we see a disruption in insulin signaling at the level of the liver membrane. We also see a reduction in the insulin receptor on the surface of the liver. And what that means downstream is that we're going to have elevated insulin that normally would be removed at the level of the liver, which is now going to be traveling downstream to, for example, the adipose tissue, skeletal tissue, to the brain, to the other peripheral organs that make up a functioning human. And if we're doing this on a chronic basis, I think one of the things that everybody agrees about in this space that's out there is that when we generate the hyperinsulinemic environment, that this is leading to peripheral insulin resistance, which is used as the argument for all of these metabolic diseases that I'm currently talking about. So what are the things that shift us from this state of burning into what I call the red state, which is again characterized, right, by this storage of fat at the level of the liver? And I'm going to focus on the liver because otherwise the conversation is going to get way too complex. Some of you may think it's already too complex, but basically the shift in this state is due to hyperglycemia above this special threshold level, the introduction of alcohol, umami, added sugars, bringing in the exogenous fructose and so on. We get the shift into what I call the red state. This is characterized by high glycemia, high insulin, and energy metabolism that is dominated by glycolytic mechanisms. That's really, really important. And the de novo lipogenesis, so the production of oil droplets, and VLDL that's high in triglycerides that are getting pushed into the circulatory system. We have other things that are going on with there. We have the insulin that is downregulating lipolysis at the level of the adipose tissue, but also there's a lactate effect there. And there's a lactate effect that's going on the mitochondria, the CPT1 and CPT2, along with malonyl. There's a lot of effects here. When we go down through a list, I think I ought to just go through very quickly so you understand why I'm arguing a change in state here, is that when we have the elevated uric acid, we have the downregulation, very important enzyme aconitase. A very important enzyme called enoyl hydratase that is involved in beta oxidation of fatty acids to produce ATP. We have downregulation of creatine kinase, which is in the cytoplasm, right? It's involved in the cyclic mechanism that takes ADP and remakes ATP from that. A very rapid cycling of ATP, downregulated when we have that acute rise in uric acid. We have the elevation and the induction of JNK1, IL-1 beta, and MKK7. All of these guys are involved in the stimulation of and the induction of systemic inflammation, which has dramatic effects on the innate immune system. Think about what's going on if that's happening on a systemic level, and it's happening chronically every day, day in, day out, week after week, and so on. And we also have the induction of carbohydrate regulatory element binding protein along with the sterol regulatory element binding protein. These are transcription factors that are involved in upregulating de novo lipogenesis, the production of the oil droplets and the triglycerides. So this is a big thing. I believe we're looking at a change in state from a burning mode in the liver to what I call the red state, which is focused on the storage, the systemic inflammation, and so on. Now, when we turn to gout, so remember that we have two things. We have the systemic fructose uric acid metabolism that is happening. This is causing the systemic elevation in uric acid. It's pushed from the liver out into the circulatory system where the uric acid can make its way all around to other organ systems and tissues in the body, including the synovial fluid in our joints. And we know, I'm not going to review it here, but we know from the literature, very, very high quality work, that when that uric acid diffuses into these other tissues down its concentration gradient, it can move from the circulatory system within the tissues on an intracellular level. And there is data which shows that once it's intracellular, it can elicit its metabolic pressures, if you will, just as if the uric acid was produced directly within that cell system. So when we look at the current gout hypothesis based on the literature going back into the 1850s, we can break the gout flare down into essentially 2 steps. And the business end of gout is in step 1. This is a situation where we have low-grade inflammation at the level of the chondrocyte. And the chondrocyte, I know this sounds technical, but the chondrocyte is the biologically important cell network with the biologically important cell, which is networked with a lot of other cell types and whatnot that are in the synovial environment. And this cell system is suffering from low-grade inflammation, which can be caused by a number of factors. The first is we know it can be caused, that there can be a metabolic shift there that is caused by hyperglycemia. And where we know this is from literally many, many papers that have been published in the area of osteoarthritis. They have looked very heavily at the effects of hyperglycemia on the chondrocyte. And they've done that because type 2 diabetic patients tend to suffer from osteoarthritis. And because of the glucocentric way that we think about diabetes, again, standing outside the forest looking in, is relative to the question of the hyperglycemia. And so there's been a lot of research in this area. And we know that when the chondrocyte is submitted to this hyperglycemic environment, that there is a metabolic shift there. And we know both at the level of the chondrocyte and the innate immune system that is present there, the macrophages, that this metabolic shift is in the direction of Warburg, which has a lot of consequences here. But I believe at the core of this, on an intracellular basis, and there is some data for this, that we have uric acid involved on the intracellular level in driving that inflammatory state. What differentiates a gouter, and I addressed this a little bit earlier when I talked about rheumatoid arthritis and gout, is that I think that low-grade inflammatory state is somewhat distinct because of our genetics, because of the gouter's genetics, and probably also with rheumatoid arthritis. When uric acid is elevated within the chondrocyte, it's going to be pushed into the extracellular environment. And from an evolutionary point of view, there was probably good reason for this. Because again, another way that we can have this metabolic shift is when there are issues with the joint from a mechanical point of view. So if you're running, you're jumping, you're doing whatever, both at the level of our hands and our feet, there will be subtle, nuanced damages to our joints. And the way that those are going to get fixed is through, I believe, and I'm going to show you some evidence for this oxidation reduction network between these molecules that signals to other proteins that are in that extracellular matrix to shift into a repair mode. And we see things that are happening at the level of genes, certain proteins that are being produced that are moved into the extracellular matrix, where that matrix can now be worked on to bring it back to a normal state so that you can move and you can do things where the joint is operating correctly. Now, where we get into trouble with this is when we are in this chronic low-grade inflammatory state 24 hours a day, day after day, week after week. And this is going to cause us a shift in this state into one which is not meant to be operating 24 hours a day, every single day. And then when we get elevated uric acid to the point where it can combine with calcium ions, we can form seed crystals, and or it is also possible that the urate is being converted into what we call urate radicals. And we know from high quality work coming out of the Kettle lab and the Miotti lab, and I'm going to move to that in a second, that these urate radicals can actually form adducts with proteins. And once they form an adduct with protein, it's conceivable that that is also a place where we could get the formation of a seed crystal. And once we have the formation of those seed crystals, then we can advance to step 2 in this process that involves the macrophages, significant number of macrophages that have internalized these crystals, maybe these protein adducts themselves. The macrophages burst, we have an infiltration of the neutrophils and the formation of nets, and we can then move to the situation where we have the hot flare, which is so painful, and in some cases can take weeks before they resolve. So it's important. I'm just showing a paper here that has shown unequivocally that uric acid can, in fact, diffuse from the synovial fluid into a chondrocyte, where it can elicit low-grade inflammation via oxidative stress. And then what you're looking at here is one of these, it's very interesting work, and it suggests not only mechanisms for gout, but a potential mechanism for cardiovascular disease as well. And this is where urate is in a specialized signaling network with other molecules that are involved in oxidation reduction, the transfer of electrons. And we know that these urate radicals exist because you can identify them once they've formed. That's number one. And I have the references at the bottom of this slide for this. And then secondly, not only do we see an elevation of uric acid in the joints of gout sufferers, but we also see the production of allantoin. We know that urate is being degraded in gout sufferers who have active flares. And we also know that urate is being degraded in people that suffer from rheumatoid arthritis, because again, you can see the elevation of allantoin that is identified in the synovial fluid from people that are suffering there. So I believe that urate, we can no longer just sit back and go, urate is just a waste product of our metabolism, and when the concentration gets too high, it crystallizes. No, urate is a signaling molecule. It's instrumental in driving the liver from the burning state into what I call the red state. And I believe it's instrumental at the level of the synovial fluid and our joints in driving an inflammatory state where you can have the formation of these seed crystals that interact with the macrophage, and also there are also neutrophils that are going to be present in that joint under conditions of low-grade inflammation. So I think that what's happening is we're crossing a threshold there. The urate rises to a point where, and when you look at this model, you can see how if one of these electron transfer agents, ascorbate, the urate, or myeloperoxidase, I'm sorry, I forgot to define that. If any one of those three things, and there are other candidates here too, by the way, from the literature, but if any of these three things are present in elevated concentrations above which they would be normally, like in response to some sort of acute injury or acute metabolic pressure, that's going to push this circuit one way or the other. And when we get the overproduction of urate radicals, for example, and have the formation or the adduct with proteins, which has been shown in the literature, then we have the possibility of generating these aggregates that could be involved in the formation of a seed crystal, interacting with the macrophage and the neutrophils and the production of nets, which also would provide an additional source of myeloperoxidase besides the membrane of the synovium, and also within the synovial fluid, which MPO exists in there, which could then get us to the second step, and where we have the generation of the flare. So in the future, if we just ignore lifestyle for a minute, which I'm going to talk about in a second, and this is the way our medical establishment looks at things. If we're looking for drugs or some sort of therapeutic intervention. Everything so far that we have that is out there is focused on the second step of the flare. We need to turn our sights on step one. That's where the business end of this is happening. One, it's a specialized inflammatory environment that might be, because of our genetic predisposition, specialized in the area of gout, perhaps happening off of the same systemic pressure that eventually can lead to osteoarthritis, or in the case of the gouter, the uric acid imbalance, or in the case of rheumatoid arthritis, which also seems to have a significant connection, as I've outlined here. So how do we get gout remission? And now I'm going to talk about the obvious things, which is to remove the direct pressures that are on the activation of fructose uric acid metabolism. So that's added sugar for sure, because that brings in not only glucose, but also fructose. And we are thinking about that threshold that I've talked about, that in my view, no one really knows exactly what it is yet. So we remove the added sugar. We stop eating meals that are hyperglycemic. And don't worry, I'm going to give you some examples. We reduce or we stop with the alcohol. And alcohol is a profound driver here. It activates the first enzyme in the polyol pathway, aldose reductase. It acutely causes the rise of lactate, which, pushed from the liver into the circulatory system, makes its way to the kidney, and then causes, because of competition issues, to be excreted into the urine, causes the backup of uric acid. So there is an acute rise in uric acid when somebody is drinking alcohol. The third mechanism is the fact that it activates xanthine oxidase, which is directly involved in the production of uric acid wherever this enzyme is being produced in concert with fructose uric acid metabolism. And the last place where it's super important is in the conversion of acetate to acetyl-CoA, which results in the production of AMP, which is then going to be degraded into uric acid via AMP deaminase. So there are four major mechanisms here that contribute to the acute rise in uric acid. And xanthine oxidase is produced within the chondrocyte. Don't forget that. We need to reduce umami, all right? And this is mainly coming in from overeating organ meats. And I'm not saying you can't eat organ meats. And in fact, in my lifestyle, I still eat them. But if you're one of these guys that's going down to the pier every day, and I'm giving you a real life example of this, somebody who I communicated with who suffers from gout. If you're going down to the pier every afternoon and you're eating oysters out the wazoo and drinking beer with it, good chance you're going to flare. Now, I'm not saying you shouldn't ever have an oyster, but you definitely don't want to be overeating them. And then the other big place where the umami is coming in, the glutamate, inosine monophosphate, adenosine monophosphate, yeast extract, for example, is going to be in the processed foods, right? All processed foods are going to be very, very high in all of the openers of fructose uric acid metabolism, the added sugar, the salt, the umami, right down the list, right? And this also goes for processed meats, because not only have you blown open the protein and you've concentrated it, right? I'm thinking of jerky, salamis, and things like that. But usually these guys are going to put in a lot of additives in addition to just that fact alone. So again, this comes down to the argument I made about the pier. Like, if you're one of these guys that's eating a ton of pepperoni every day, that's going to have to be pushed back on. Processed food is not a good thing here. You want to shift to a whole real food environment. And this is one, And I'm not going to apologize about it. This is an eating program where you buy real food that looks like food, you cut it up, and you cook it yourself. You get into a mode where you produce enough food every day so that the next day at lunch and whatnot, you're eating leftovers that are actually made, from whole food. You avoid the processed food trap. This includes going out and eating in restaurants. Restaurant eating should be selective and be done carefully. Why? Because everything that you eat in a restaurant is going to be processed. And essentially, you cannot trust those guys that the additives, the things that they add to that food and the sauces and whatnot are going to be done thinking about you, thinking about, all right, this guy who's going to eat this suffers from gout. So I better not add the added sugar, and I better not put monosodium glutamate, shaking monosodium glutamate into this food or mixing it into the hamburgers that they make, right? You are best if you buy whole food, cut it up, and cook it yourself. If you suffer from a comorbidity in addition to the gout, like it was in my case, for example, I had the gout and I had the prediabetes, then you're going to need to cut in, not only make sure you've eliminated all the things I'm talking about, but you're going to want to cut into the carbs. And I'm not an influencer who's going to get out there and make the argument with the establishment. I think it's a really silly argument. Everybody knows, who's ever really encountered this. And you can look at Sarah Hallberg's classic TEDx video if you want. You just cut into the carbs and get yourself keto adapted. Monitor your uric acid, right? Be prepared that you could flare. This is something that in my space, the low carb doctors and whatnot will argue against. They'll say, well, there's no risk really, and you could possibly flare, but usually it doesn't happen. But for 5 years, I've worked with gouters, and I know that when you go low carb with it, that there is a fraction of us that will suffer from gout flares. We will. So be prepared for that, and make sure to have a lot of anti-inflammatories around, and make sure to have a medical doctor that you're going to and you're talking with, which could be a challenge to find one of those guys that's willing to work with you, but you need to do it. It's really smart on this whole food shift in the lifestyle to make sure to vary the types of natural proteins that you're eating. I've run into a lot of guys over the last 5 years, and I mean guys, male or female, who are tending towards carnivore, and they're eating the same thing every single day, like lots of ribeye steaks, morning, midday, and dusk, and suffering gout flares. It's best when we shift to this whole food environment to make sure to manipulate the types of proteins we're having, because your body metabolically adapts to things. And well, I don't agree with the argument that you should exclude protein from your diet in order to be gout-free. I don't believe that that's true, especially when you look at the literature going back into the 1850s. Those guys found that eliminating protein was not, it didn't result in people not having gout flares. So I think having protein is a good thing. I think that you should vary the types over the course of the week and not get into a thing where you're eating the same type of meat every single day. In the beginning, it's smart if you shift to a meal plan where you're having, say, 3 meals a day. And if you're cutting into the carbs, it won't be very long before you will be down to eating probably only 2 meals a day. Now, fundamentally, will probably be related to whatever your activity level is, and I'm going to talk about that in a few minutes. If you're like me and you're highly athletic, I'm still eating 3 meals a day because I need the calories, quite frankly, in order to do the things that I do. And I'm a CrossFit athlete, and I'm rock climbing outside 1 to 2 days a week, and I'm in the rock climbing gym, and I'm throwing weights around and doing stuff like that. So for me, in my lifestyle, I'm eating more than 2 meals a day, although I do measure my ketones still, and I am in mild ketosis. And then make sure to vary the fats. And I'm not going to be one of these guys that's going to argue over whether you should be having olive oil and avocado oil as the main oils all day long, every day. I eat a range of different natural fats. I stay away from the seed oils. I stay away from the oils that are refined for sure. This is a really smart thing to do. And I feel about the oils and the fats the same way I do about the protein. Modulation. Having a lot of really good natural fats in your life is a good thing. Beef tallow, lard. I collect my bacon grease, for example. I use it all the time. Ghee, avocado oil, the olive oil. Down the road we go on coconut oil. I think having a range of natural oils is a really smart thing. Those of you that are asking me questions right now, I'm not ignoring you. I just want to finish the presentation and then I'll answer questions. Okay, and I know this has been a long presentation, but it's just where I feel I needed to go because I've been away for a while, and I think it's good. So right now you're looking at a slide that over the last 5 years, over and over and over again, I think one of the things we hear from the pushback that we get about all this business is that I'm not eating much added sugar. And yet when I get with people and I talk with them, electronic forums, extended family, friends, you're looking at what I feel is a really good representation of how they're eating. And all of these are incredible gout foods. But not only that, they're driving diabetes, the obesity epidemic, cardiovascular disease. Down the road we go. And the one on the lower left that I'm showing there, the pancakes, the way that got on this slide is that I have extended family that think eating a nice big pancake breakfast is good for them. Look at the berries that are on there and the syrup that's streaming down those refined carbohydrate pancakes. Boom, hyperglycemic out the butt. That thing, I guarantee you, was well above the threshold to activate polyol pathway in the liver. The top meal, I'm picking on that because I can think this is the way I used to eat, right? I can remember going into my favorite pizzeria here in Phoenix area and ordering a large salsiccia pizza. I would have 2 or 3 beers during the meal, along with a salad with that came with high glycemic dressings and so on. And the thing is, I used to tell myself, well, this is special tonight. I don't eat this way all the time. But truth of the matter is, when you sit back and you think about what these guys are eating, the way I was eating at breakfast and then lunch with the morning snack and the afternoon snack, and then a meal that was, would you look down at the plate and not that different than the one down there on the right. Look at all of the potatoes there, the biscuit, right? You probably put a bunch of butter on the biscuit. That biscuit is like on its own merit would just slam that hyperglycemic threshold. And then I used to like the sushi, and in there, if you look closely, there's sake with that, and there's rice for the hyperglycemic effect. The soy sauce not only had sugar in it, but I used to add extra salt. Salt opens the polyol pathway, especially in the presence of these other things. And it's like, all right, this is an example. Let's just get to a grounded place where how it is that we're eating out there on a daily basis, right? Here's more examples, right? The one on the lower part there, I have specific experience with in terms of clients. There's the full glass of Coca-Cola at 41 grams of refined table sugar or high fructose corn syrup, depending on the source. Look at the buns on the burger, and then we've got the fries, the hyperglycemic fries that were done in a deep fryer with seed oils where that oil probably hasn't been changed out in months. This is just another example of the way people are eating lunch. I've got the beer in there again. We all hear in gout about the beer, and that's true. And the beer isn't just bad because of the alcohol. And I already talked about 4 effects in alcohol. It's got the purines in there. And also, generally speaking, they're very high in carbohydrate. So while you're waiting for the meal and you have 2 or 3 of these beers, I can remember doing that. You're probably already close to 75 grams of refined carbs, not even talking about the purine or the alcohol effect before the meal even shows up that you can see here. And there's even a breaded onion on that burger in the upper right hand corner. And these are the typical breakfasts that I've actually seen over and over and over again in my extended family. There we have on the left side the low-fat yogurt with those big fat strawberries, right? That's nothing like at all the way strawberries actually grow in the wild, right? These things are Mongo. They've been bred for high sugar content. And then what else did they? Oh, they put some heart-healthy oats in there. Okay, boom, hyperglycemic polyol pathway activated. Fructose uric acid is rocking and rolling in the liver with a breakfast like the ones that are shown here. No question about it. Don't stop, go. This is rocking and rolling. So now we're shifting to the whole real food environment. And, if you look at these plates, the first thing that you're going to see is the absence of the hyperglycemia. This is the way I eat every single day. These are my meals that you're looking at. And if we break the plates into 3 segments, one segment has protein in it. And then 2 of the segments have low glycemic vegetables that are either roasted or steamed or made in a cream sauce that I make myself. I would never buy a cream sauce in the store. Why? Because there's going to be unhealthy fat in it. They will have added sugar to it, and it will be BAMO for umami additives like monosodium glutamate. For example, on the right, you can see that's a pork loin roast that I made. There's yellow squash there and roasted Brussels sprouts. Even if you track these meals using a glucometer like Keto Moho, these meals are not going to raise your glucose into the range that's required to break that threshold and open polyol. And uric acid, you will see uric acid go up after these meals. Of course, you're going to see it go up because there's protein in the meal. I'm not arguing in this presentation anywhere through here am I saying that when you eat protein, you're not going to see uric acid go up. Normally, it should go up. And I think a better argument for the fact that it goes up is because uric acid is a signaling molecule. What does it signal? It signals storage capacity. So from an evolutionary point of view, you expect it to go up when you eat protein because that is driving a state of repair, a state of protein synthesis, and so on. I don't have time in this presentation to get into those arguments, but I think that is a much better argument. The question is, how high is it driving the uric acid? And I don't think we should be concerned about uric acid elevations that are normal functions of a human, in part when we're eating protein, and secondly, when we're doing exercise. These just show you my typical breakfast. There's lots of ways to have breakfasts, and you'll notice that I have some sort of protein source along with my eggs every day, and I eat different kinds of eggs. I've 5 years in this lifestyle. I'm in gout remission, and I reversed my diabetes, and my biomarkers look pretty good. Additional interventions. We can't just talk about the food here. There are issues with sleep. Hypoxic environments are normal in all of our organs and tissues. And in sleep, it's typical for people that are suffering from metabolic disorders to also be suffering from sleep apnea. I know that in obese individuals, that 50 to 80% of them are also sleep apnea individuals. And every time you have an apnea event, which can be many, many times within an hour during the sleep cycles that you are causing extreme elevations in hypoxic environments because you actually stop breathing during those moments that you're apnea. And the problem with elevating the hypoxic environment is you cause this metabolic shift, which moves towards Warburg and is part of the fructose uric acid metabolism, which causes this acute rise in uric acid, which is part of the signaling network, which is an oxidation reduction network, which causes a shift in different states in our cellular systems. And we do not want to be in this chronic elevated state of uric acid. So if you are listening to this and you suffer from gout, get sleep tested right away. You need to do that. Even if you think for whatever reason that this isn't you. I got sleep tested and I am not a regular snoring person. And it turned out, in addition to all this other business with the gout and the prediabetes, that in fact I had mild sleep apnea, and in fact I am now treated for that, and I've reversed the apnea. All right, exercise, super important. All of us need to be doing it. I don't want to take the time in this video to get into the details about it because I already see that I've been going well over an hour. And then the issue of stress. Yes, some of you are listening to this and you're going to go, Dr. Pete, what about the cortisol effect? Yeah, there is one. I haven't had the time yet to beat that to death, but doing something about the stress in your life, relationships, the stress at work, trying to come to an understanding how to modulate that so that you can bring the stress down is super important to becoming gout-free. You can measure biomarkers, right? 5 years ago, everyone in this space was telling me, you don't need to do that. Just eat low carb and everything's going to be better. Have faith in the way that you're eating. Nowadays, everyone is on the train about getting a CGM, a continuous glucose monitor. All right. I think it's really smart if you measure your biomarkers because you can know your glucose and your ketones and your uric acid before you eat a meal, and you can know what those values are after eating a meal. That will allow you to tweak what's on the plate. All right. And you can even see there's a lactate meter on here. And that's because I've been doing a lot of N1 to try and understand what the relationship is between my glucose, my ketones, my uric acid, and my lactate fluctuation, both before and after meals, before and after alcohol, and additionally to before and after exercise, to understand what my cycles look like. And the reason lactate is cool is because it gives you a way to try and understand different modes of exercise and how you can apply that to improving your overall fitness. Outcomes. Shifting in this lifestyle, you get gout remission. You can do it. I know you can do it because I've done it. You can reverse diabetes. I know you can do it because I've done it. And then, by the way, in number 1 and 2 here, well, actually all of them, I have actively coached people now for 5 years. And I've had people that have stage 4 kidney disease that I've worked with who have improved their kidney function. I've seen the reversal of type 2 diabetes with my coaching frequently, right? Every single client I work with can do this. It's not, this is not out in left field, and the gout remission and so on. And anything that is centered around this central mechanism of fructose uric acid metabolism will get better if you eliminate the main drivers. And with that, I'd like to thank you for your attention today. You can find me at peterdelanoy@drpeteandT.com. That's my email directly if you want to ask questions. We have a website, www.drpeteandT.com, that talks about our generalized programs in order to help people reverse these conditions, including sugar and alcohol addiction. I might be the only one in the low carb space that's done a 30-day live-in with a sugar addict and successfully put them in recovery, reverse diabetes, and they lost 11 pounds of weight. And then lastly, if you're actively trying to reverse gout, my website for that is www.drpetesgoutintensive.com. All right, so you can reach me there. All right, now I'm going to go ahead. For those of you, I want to thank you guys for being here. And this has been a really long presentation. I'm now going to turn to the questions. Let's see. Okay, methylene blue and light therapy. I don't know anything about that. Yes, this is right from Phil. Drinking allopurinol and drinking lots of water. Yes, those crystals will go. You can also supplement that with potassium citrate. That will reduce the excretion effect, which it increases the excretion effect. It reduces the possibility of a kidney stone. Okay, is it possible to bring down serum uric acid without allopurinol? Yeah, it is. You can take glycine. And this has been shown in the literature. It was a clinical study. I don't know why we never hear about it. 12 grams of glycine per day. But if you're going to do the glycine trick, it's crucial to also do potassium citrate, because the fact is, once you start the glycine, you're going to increase the excretion of uric acid into your urine. And those of us that gouters that trend towards that specialized inflammatory environment with the formation of seed crystals seem to also have a predisposition to formation of kidney stones. So if you're going to do the glycine, also do potassium citrate to raise the pH of that urine and increase the solubility of uric acid to decrease the probability of getting a stone. I agree with the allopurinol, but some of us have side effects to it. And this recent data that comes from the Kettle laboratory, which is really amazing, high quality work, shows that that allopurinol, that there's a sweet spot with it. That if the dosage gets too high, it actually can increase oxidative stress. And I believe that perhaps passing that threshold is the reason why some of us experience side effects with allopurinol. So if you're going to do the allopurinol, I actually think a smart thing to do is to keep the dosage as low as possible, right? Also coupling that with glycine and potassium citrate, so you can bring the allopurinol down lower, still getting the optimum lowering of the uric acid, but without crossing that threshold into the point where the allopurinol is actually causing oxidative stress because of the production of oxypurinol. I agree. Okay, so if I didn't say it already, everyone here ought to be under the care of a doctor, and you need to be talking to them, and finding the optimum dosage of allopurinol is, something that you need to do with your doctor. I'm not a medical doctor. I'm not giving you medical advice right now. I think what it is that you really need to figure out the minimum amount of allopurinol that you need. And you can, the amazing thing about the glycine is I think you have a way of doing that now, because you can bring the glycine up safely. I can think of no negative reasons to manipulate the glycine. There's none in the clinical study that was done. Make sure that you have the potassium citrate effect in there, right? To keep the urine pH elevated, to decrease the chance that you're going to get a kidney stone. And I think you can do this, right? And I think You ought to do it. I don't know the answer about the artificial sweetener thing, but sorry about that, guys. I don't know the answer about artificial sweeteners, but I'm going to caution you about their usage for two main reasons. The first is that if you have a sugar addiction problem, which is quite often found with gouters, all right, this is my experience coaching directly with them. There's almost always an issue giving up the sugar. Artificial sweeteners can track you back in to arrive back at using sugar over again. So I would be really careful about that from that point of view. And then the second thing is, we know for sure that the sugar alcohols in these non-sugar substitutes are modulating changes in the gut biome, and we know that the gut biome is involved in gout. We know it. We just don't know specifically what's going on with that. So I try not to eat anything that I know is messing with my biome. And along those lines, I think bringing fermented foods like sauerkraut and well done the non-standard dill pickles, things like that, without going extreme, bringing those into your lifestyle, I think are probably a good thing for the microbiome. Okay, I think I answered all the questions. Okay, this top one, I think I ought to talk about just briefly over again. If you're eating protein, it's gonna cause a rise in your uric acid. If you are actually measuring biomarkers and you get yourself a Urate 2 meter, and I know it's expensive and all that, but you can follow what the rise in uric acid is going to be for a particular meat that you're eating. And then if you choose, you can modulate the amount of that meat, right? Let me give you an example. If you eat liver, like I've done this experiment on myself, if you have a sample of liver, you can guesstimate, okay, I'm going to put some of this on my plate. Measure the uric acid before you eat it, and then measure the uric acid at a half an hour, hour, and 2 hours after you eat it. And you're going to see your uric acid come up. And you can make a decision on that. All right, either, wow, it really spiked me, I'm not eating that stuff. Or, okay, this is only causing, it's only a small amount that my uric acid came up. It's down within 2 hours. I could add a little bit more liver into my lifestyle now. I think I can do that. So you can learn to gauge how much of this is a concern or not. And honestly, I'm eating these days with a lot of extra protein in my diet because of my exercise regime. And I'm using allopurinol coupled with glycine, potassium citrate, to keep my uric acid down around 5. And I'm going forward right now in that mode. So that's where I'm at with this. Now, I think when I look at this, I don't see any more questions. I want to thank all of you guys for being here today. And I'm going to be now posting again regularly. I did take a break from this, but again, thank you very much. And I would like to, I'm going to go ahead and close now. You guys all have a great day, and I'll see you the next time I'm here. Thank you very much.